So one quick thing, I don't want to go into too many details because I don't want to run out of time, but I didn't get to it last time. You know, the discussion boards, we have one last discussion board for this unit, and it's immune system web links. I want you guys to share stuff you find on the internet. I noticed earlier it had an earlier deadline, and I just changed that to November 6th. Remember, my deadlines aren't really hard, fast. I'm not evil like that. So everybody always has some excuse. Just get it done. As far as discussion boards are, I have to physically grade those, right? So chemicals I forgot about, I apologize. I will get on that as soon as possible. But as long as you follow the instructions, just like for the introduction discussion board, you should get all your points. And there's a rubric, right, which is how I grade and how you get your points, which is basically just the instructions, right? And so if you lost points, like for instance on the introduction discussion board, you might want to look at your grade and look at the rubric. There's a little icon there. Or look at any comments I may have wrote you. At this point, if I gave you a second chance back then, it's too late now. So, but be looking for that for chemicals once I start grading that, right? Because I usually will sometimes let you guys fix a mistake. But you got to do it within a timely period, right, in order to get those points. Okay. So I just wanted to mention that. I added it also to the module. And so for this module, we're just covering 2 chapters, right? We finished chapter 19, so soon I will also have a Wiley Plus assignment for you. And then we're on to the next today. We're actually on to I didn't even open my stuff yet. We're on to chapter 20, which is adaptive immunity. And I posted the objectives list as well as the first part of the lecture last night instead of this morning. So the opener, if you read for this chapter, is kind of frightening. They talk about bioengineering, basically a virus, yeah, they made a really lethal virus, and weren't planning on doing that. So it's definitely one of those things to think about when we start tinkering with these things. So in this chapter, right, we're going to start out today by talking about the features of the adaptive immune response. Then we're going to really focus in on T cells. We're going to talk about antigen processing, and then antigen presenting cells. And I mentioned these previously, right, in our other lecture. Even within adaptive immunity, we kind of divide it into 2 subsets, what's referred to as humoral and cellular immunity. And one of the things to keep in mind, too, is these aren't necessarily completely separate from each other. Just like we talk about innate versus adaptive, these work together, right? We've already talked about some of the components of adaptive immunity and how they work in conjunction with innate and therefore vice versa. And then the last thing we're going to talk about is my favorite thing, is B cells and the production of antibody, right? So antibodies are pretty amazing things. So we're going to start out with what is adaptive immunity? And then, of course, how is it different from innate? So why do we have these 2 distinctions? Well, remember, adaptive is only found in jawed or higher vertebrates, right, like ourselves. So invertebrate, lower level animals don't have this extra layer to their immunity. They just have innate. Remember, innate is you're born with it. All that information and how you utilize it is inherited from your parents. The difference for adaptive, one of the major differences, is that a lot of times innate is referred to as non-specific. Adaptive, on the other hand, is very specific. There's a very high level of specificity for specific molecules on the pathogens that we are defending against. And that's important because that actually literally will allow us to distinguish one bacteria from another, one virus from another. So remember, as I said, innate, non-specific, and it relies on patterns, right? Recognition receptors. So these very common patterns that we see that say it's foreign, right? That it's bad. So can you guys remember some of the things that we have receptors for? So what are found on microbes that we can identify in a general means? So what are some substances that are associated with, say, maybe gram-positive bacteria or gram-negative bacteria? LPS, what does that stand for? Lipopolysaccharides, right? So lipotectonic acids, right? Lipopolysaccharides are the outer covering of the outer membrane of gram-negatives. Gram-positives have lipotectonic acid, and then their cell wall is made of a very thick layer of what? Peptidoglycan. So we have receptors for things like that. Remember, we have receptors for mannose, right? So these substances, does that say that, you know, it's E. coli that got in? If we have a receptor that's recognizing LPS, does our body know, oh, E. coli infection? No, they know it's a foreign invader in this classification. And they're going to generally respond. Where the difference for adaptive, they can specifically, even down to the different strains of E. coli, discern one from another. Then, so not only we very specifically recognizing the pathogen, right down to its molecular level, different sugars on the surface of that bacteria, but we remember these encounters, right? So our system has a memory function in it. So when you're exposed to something once, you remember that exposure, right? So say you're a little kid and you put your hand on a hot stove and you burn it. Hurts like hell, right? The next time you come by that stove, right, you going to touch it again? No, because you remember that it burnt you. At least you hope you do, right? Most of us learn that way. And our system kind of learns that way too, right? So it recognizes something as being bad and it remembers it. And it responds accordingly the second time, right? This is bad. I need to take care of this problem. Does that make sense to you guys? So do you guys know anything that we can do that helps with this memory function? What do we do around this time of the year to get our immune system to develop a memory response? The flu vaccine, right? So they're giving you the virus, right, or the viruses, in a form that it does not replicate, right? So you cannot get the flu from the flu vaccine. Anybody who tells you that, tell them to go read the CDC's website, right? They don't believe it. Yeah, they think it's a hoax, but it's true. They're not giving you an intact virus. It cannot replicate, okay? But it is information, right? It's like a wanted poster, right? You're showing your body, hey, this is the bad guy. Get prepared in case you see him again. Make sense? Okay. And the reason why we can do that is because of this memory component. And so how do these cells actually interact with these foreign substances we call antigens? Well, I alluded to this last time, right? T cells have receptors on their surface specifically to interact with antigens, okay, foreign substances. Notice these B cells, this receptor, does this one look the same as this one and this one? No, each one of these B cells, their receptor looks different, right? Why does it look different? Why wouldn't they look the same? They're fighting different things, right? This T cell binds to something different than what this T cell binds to. Each one of your T cells, when they're made in your bone marrow, they're different from each other. their receptor is different, because how many different antigens are there out there? Millions upon billions, and they're always changing, right? Viruses change, bacteria change. So if we don't make different T cells with receptors to recognize those antigens, then we're going to have no hope of recognizing them. But this is what's one thing that's really interesting about this system, is that we make the T cells even before we see the antigen. So they don't make these receptors to fit what you get invaded with. They just make T cells. So this is like if you want to win the lottery, you buy like a gazillion lottery tickets, right? So we buy like a gazillion lottery tickets. We make T cells and B cells, we make millions upon billions of them, all with different receptors, with the hope that one of those B cells or one of those T cells will recognize the foreign entities that enter into our body, that we get a match. Right? And that's the awesome time, right? In the case of the lottery, you're like, yeah, I have a ton of money. Here it's like, okay, now I have protection. I know, I got this guy's number, right? I know him. I can recognize him. So for T cells, it's a T cell receptor. It has just one antigen binding site. So they're showing this, opening here. This is where the antigen would bind to this T cell, right? And they would recognize each other. For B cells, the B cell receptor is actually an antibody, right? And again, notice the difference. For these guys, they have two antigen binding sites, right? This antibody has this characteristic Y shape. So it has two antigen binding sites. And notice again, this B cell is different from this B cell and different from this B cell, right? Each B cell is unique when it's made. The difference between T cells and B cells is B cells, when they become activated, can actually make this antibody they have on their surface or a different class of antibody, and they can actually secrete them. So they can go into our bloodstream, they can travel throughout the liquid of our body, right? So they'll go through the bloodstream, we put them in our mucus, they'll go into the tissue when we have inflammation, right? Anywhere you have where these proteins can go, they will go, right? And the great thing about them is they can bind to specific antigens, right? So they're literally actually going to bind. So as I said, the immune receptors bind to antigen, right? So these foreign substances, and we're going to define antigen in a moment. T cells have T cell receptors. B cells possess immunoglobulins, right? Remember I said last time that's another name for an antibody. Immuno referring to immune system, globulin referring to the fact that they're actually proteins, right? So the B cell receptor is when the antibody's on the surface of the B cell. It's actually embedded in the membrane. When it's secreted, we refer to it as an antibody or an immunoglobulin. So what do I mean when I say antigen? Antigens are what will bind to these receptors. So this is a very specific term. So T cell receptors, B cell receptors, immunoglobulin or antibody. If that substance binds to it, it's referred to as an antigen. So in Chapter 19, we talked about substances that bind to receptors, and we refer to them as ligands, right? So that's general. For most receptors, whatever binds to it is generally referred to as a ligand. Another general term that you'll hear is immunogen. And this is a term, again, referring to something that the immune system is recognizing, that it's mounting an immune response. So, this could be LPS, right? Lipopolysaccharide. That's a foreign substance that we have receptors for, right? We have pattern recognition receptors for, toll-like receptors, right, that will bind to that. That's not a B cell receptor or a T cell receptor or an antibody binding to it. We could make those against it. It could be an antigen as well. But when we say antigen, we're being very specific in that it's something that binds to either a B cell receptor, T cell receptor, or an antibody. Does that make sense to you guys? All right, so it's a very specific term where ligand and immunogens are more general terms. Then we can get even smaller in our talk about antigens, because antigens tend to be very large. And it's actually a smaller portion of the antigen that actually interacts with the receptor or the antibody. That small portion of the antigen is known as an epitope. And so we'll look at a picture of that in a moment. Because epitopes are so small and antigens themselves can be relatively big, so think of an E. coli, right? We could make an antibody against a sugar complex on their cell surface. We could make an antibody against maybe a protein structure on the surface. We could have a T cell that recognizes products of when we break down that bacteria, and they're presented by other cells. So when we look at this, we thankfully, you could say, don't have to have just one lottery ticket that matches, right? Instead, it's a whole bunch of numbers, right? And so we may have a bunch of antibodies or a bunch of T cell receptors that recognize a particular antigen. Okay, so B cells can actually directly interact with antigens. As we'll see later next time when we start talking about T cells actually have to have antigen presented to them by other cells of our body. But B cells can directly interact with an antigen. So remember, their B cell receptor is actually a membrane-bound antibody. So notice this antigen, whatever it is, maybe it's the flu virus, right? Maybe it's E. coli 0157. It has lots of different molecules on its surface. So notice this molecule, they're denoting this shape, another molecule over here, another one. Each one of them has its own unique signature, right? They're showing different shapes and colors here. But we could be talking about proteins that are made up of a certain set of amino acids, or sugars complexes that are made up of certain sugars. So this interaction, y'all, is down to the molecular level, where these antibodies or B cell receptors are binding to sections like 10 amino acids on the surface of an antigen. Right? So even a protein itself can have multiple antibodies bind to it, because they bind to such small molecular sections on these molecules. And because the molecule isn't all completely the same, right? You don't have a protein even that's made of nothing but phenylalanine, do you? No, you're going to have a mixture of different amino acids in different combinations. So you have, the potential of numerous antibodies being able to bind to that one antigen. So here they're showing, right, here's the B cell interacting with this specific epitope, right? This little piece, molecular piece of the antigen. Where this different antibody, right, noticed by a different shape to its antigen binding site, and the color of the antibody they even made different, to really show you that this antibody is different from this antibody, and this antibody that's the B cell receptor because it's attached to the B cell. But they're each binding to something different. So your response, even to something like a protein, is not going to be just a single B cell recognizing it, or a single antibody. You're usually going to produce multiple antibodies against multiple different sites on that antigen. Those sites that actually interact with the B cell receptor or the antibodies are known as epitopes. Sometimes you'll hear them referred to as antigenic determinants. most widely used as epitopes. So as I said, this is a very specific molecular interaction between these receptors and that antigen. So they're recognizing it. They're literally just recognizing it. Yeah. Right. The first step, right, in being able to eliminate an invader is recognizing it, right? Recognizing that this is foreign. This is not me. So this is key. The key thing about this, though, is, like I said, the level of specificity that we can get here, right? We're not just binding to LPS. We're binding to specific, maybe, sugars found on a specific strain of E. coli. So it was a very specific interaction. Very targeted attack and recognition. So B cells and T cells, which are lymphocytes, right? So sometimes they're referred to as B lymphocytes and T lymphocytes, but a lot of times they're just referred to as B cells and T cells for short. All your white blood cells are made in your bone marrow, right? So these are white blood cells, right? So they're made in your bone marrow. But these differ from like your neutrophils and your macrophages and your dendritic cells and all of that, and that they have these antigen-specific receptors on them when they're made. And each one of them is made differently. The B cells are actually named after an organ in a chicken called the bursa, where they first discovered B cells. But luckily enough for us, bone marrow also starts with a B, right? Bone starts with B. When these guys leave the bone marrow, they have their receptors, their B cell receptors on them, and they're what's referred to as mature B cells, but they're naive. They've never encountered antigen yet. And they travel all throughout our body, and they'll tend to collect in particular organs in lymphatic tissues throughout our body. So they congregate in large numbers in our lymph nodes. They travel through our lymphatic vessels. There are patches, right, known as prior patches. You can actually see them under the microscope when you look at the intestines. Because remember, our intestines is this big long tube that goes through our body. And just as long as if something goes in and right back out the other end, no problem. If it escapes out of the intestines into the body, that's when we run into problems, right? When it breaches. So within the layers of the intestines, you have very large concentrations of lymphatic cells, of B cells and T cells, macrophages and dendritic cells. So that way, if anything gets across, we're right there ready to defend, right? We don't need necessarily an inflammatory response unless we've got a large invasion. And in fact, your small intestine samples the stuff that goes through your gut all the time. It's like, okay, let me see what this is. They literally will pull stuff in, right? Check it out so they can build up immunity to it. So your tonsils, again, those are very large concentrations of white blood cells, mostly lymphocytes, B cells and T cells, and macrophages, right? Because you got to have something to eat the bad guys, right? And that's what your macrophages are going to do.
Question.
Mm-hmm.
Your body is always recognizing new organisms and building up a
immunity, memory response.
Where is this immunity memory response located at?
So it doesn't necessarily stay put. So if the response happened in a particular lymph node, you'll have a large concentration of memory cells there, but others will travel throughout the body.
And tell it.
Yeah. And again, when I said we were going to talk about the immune system in this class, and then we would just really kind of be skipping along the surface, we weren't going to go into a lot of detail. Even if you're in a graduate level immunology class, there's some of these questions you're going to want to ask. We don't know the answer yet. We have a general idea, but we don't know for sure. There's still, it's still so much of a mystery. It's amazing. It's a really amazing system. So B cells, right, are going to be born from our bone marrow, right, travel throughout, and they're going to really congregate in those areas, in the Peyer's patches, in your spleen, in your lymph nodes, in your tonsils, in your appendix. All along your skin, they congregate in areas as well. So you have what are referred to as skin-associated lymphoid tissue, where along in your skin, right, there's these collections of B cells and T cells and macrophages. And same thing in your gut. A lot of times those Peyer's patches are collectively referred to as the mucus-associated lymphoid tissue. So these guys are all throughout the body, and there are areas where they tend to congregate, which are areas where stuff gets in, right? So your tonsils help protect your nose and your throat, right? All the stuff that you breathe in all the time, right? They're right there to help trap and defend. And then all along the small intestines, your spleen, it's actually the only organ you can live without if it gets damaged and they have to remove it, because it doesn't have a vital function to life, but it does filter your blood for pathogens. So if you don't have a spleen anymore, you don't have that level of immunological protection. You don't have something that's filtering your blood all the time. No, not necessarily. I mean, you have lots of protection, but you're more apt to be vulnerable to stuff that gets into your blood, yes. Now, it also recycles your red blood cells, but your liver takes over that job if you lose your spleen. And in fact, when you're first born, your liver takes care of most of that, getting rid of damaged old red blood cells. So the T cells, though, are named after a different organ that we actually do have. They're named after the thymus. Because when they leave the bone marrow, the first place they travel to, though, is your thymus. Right? It's located right here, right above your heart. It's a glandular gland. And this is where the T cells go to learn the difference between self and non-self. Because you do not want T cells, because remember, we randomly make these receptors. What happens if we make a T cell receptor that recognizes our own cells? Do we want that? No, because then it's going to tell the system, hey, this is a bad guy. No, it's not. It's my cell. Please don't kill me. Right? So we need to have a way of eliminating those cells. And then we also have two different subsets of T cells. So this is a lot of work. So this is done by a whole separate organ. Unlike the B cells, they're checked out before they leave the bone marrow. For the most part, right, unless we make a mistake, we're not going to let any B cells out of the bone marrow that recognize self-antigens. We're going to eliminate them in the bone marrow. T cells, on the other hand, have a little more complicated process, and they travel to the thymus. And that's actually where they get their name, right? That's why they're called T cells. That's where they get their, you could say, secondary education. And so the bone marrow and the thymus are a lot of times referred to as your primary lymphoid organs, right? This is where your lymphocytes originate from, and this is where they're educated. When they leave those organs, they're mature B or T cells. But they're naive until they actually come in contact with antigen. And they'll usually do that in the secondary lymphoid organs, like your tonsils and your spleen, right? Or in the lymphatic tissues, the Peyer's patches and such. Or just about anywhere in the body where they encounter antigen. How is it that we make all these different T cell and B cell receptors? You have in your DNA how to make these receptors. But if we just use the DNA in the form that it is that you got from your parents, all your B cells and all your T cell receptors would be the same, wouldn't they? But we don't have enough space to have a whole bunch of genes to make a whole bunch of different receptors. One, that would take up too much space. Two, again, we wouldn't get variability, right? Because we'd just be copying the same things over and over again. So instead, what happens in these cells is they literally rearrange their DNA. They pick segments in the DNA and they put them together and they cut out other pieces. So it's like when you pick your lottery numbers, right? You've got, I don't know, I haven't played the lottery in forever because I know I'm not going to win. How many numbers do you have to choose from? I don't even know anymore. Like if you're doing Powerball, how many numbers is it?
80 million.
No, but I mean like on the sheet, what are the numbers 1 through what you have to choose from? Only 6 numbers? No. Like is it 40 numbers? Like is it 1 to 40, right? Is it like 1 to 40? Let's just say it's 1 to 40, right? So you have 1 to 40 and you have to pick 6 of those numbers, right? So the same thing, imagine your DNA, right? You have 40 pieces and you've got to pick 3 from those 40 and put them together. So, we end up with all these multitude of possibilities, right? I can't even think about like if you had 40 numbers to choose from and how many, but to give you an example of one I know by heart, right, is that for DNA, right, we have, when we get down to the actual code, we have 4 different nucleotides, right? Adenine, guanine, cytosine, and thymine. AT, and G and C, right? That's the code. And that code is read by our ribosomes in groups of 3 nucleotides, right? You end up with a possibility of 64 different combinations. So just with 4 possibilities and getting to pick 3, you end up with 64 different possibilities. So think about it. If you had, I'm not a math teacher, I can't do this in my head, but if you had 40 numbers to choose from and you get to pick 6 of them, how many different combinations do you think there are? A lot, right? Definitely a lot of different, does that make sense to you guys? Lots of different combinations. And this is what makes each one of them unique. And as I said, we do this before. I mean, that'd be really easy to do, right? If you picked your lottery numbers after the lottery numbers have been pulled. Oh, yeah. No, it doesn't work that way. It doesn't work that way for our body either, right? They put together their little receptors. They basically pick their lottery numbers, and they hope they win, right? And you're constantly doing this, right? You're constantly making these receptors in the hopes that one of them is going to fit, or multiple ones are going to fit the pathogen that came into your body. We just, yeah, and we're going to keep trying until, it wins or we win. It depends on what medicine you're talking about. All right. Does everyone understand this concept? This is important, right? So each one of your B cells is unique. The receptor, each one of your T cells, and that's because it's literally rearranging your DNA to make a different receptor for each B cell and each T cell in your bone marrow. Right? So when these guys are born, each one of them are beautifully unique because they literally rearrange the DNA. So once mature, right, they're expelled into the peripheral bloodstream as mature, as I said, naive lymphocytes. They're mature, they have their B cell and T cell receptors, and they're going to travel all throughout the body, where they're going to encounter the bad guys. So one other thing that they show you in this picture is that notice she has these green vessels, right? These are referred to as lymphatic vessels. These are not blood vessels. These are lymphatic vessels. And unlike the circulatory system, it is not a circular system, right? It starts in the tissues, and all those vessels combine into several trunks, and then finally into 2 ducts that dump into your subclavian veins, so right up here under your clavicle. You have the right and left lymphatic ducts, right? So all the lymphatic vessels come together, and all that fluid that leaked into your tissue when you had inflammation gets sucked up by your lymphatic vessels. And all along the way, they encounter lymph nodes. Lymph nodes are basically filters for lymph, which is the name of the fluid that goes through your lymphatic vessels. So that fluid that leaked into your tissues during inflammation, we're going to soak it up and we're going to filter it several times until finally we're going to dump that filtered, hopefully clean, liquid back into the circulatory system, right? Because fluid is very important, right? As you know, if you've got low pressure in the pipes, there's no water that comes out the shower, right? You're like standing there and it's dripping on your head, right? We don't want there to be low pressure in our circulatory system. So we recycle that fluid back into the system. But why don't we just put it right back into the blood vessels? It could be contaminated. So notice I said we filter it, we hope it's clean. So remember, what's in those lymph nodes? B cells, T cells, macrophages. So anything in that lymph is hopefully going to get trapped in those lymph nodes and be dealt with. And then you might have a B cell in there that recognizes it and starts producing antibodies, and it reproduces itself. And those B cells and those antibodies will travel through that lymph and eventually get into the bloodstream as well and travel all throughout the body to help protect you. So they travel, right, all the time. They're traveling around. So we talk about when we're exposed to a particular antigen, the primary response versus the memory response. And a lot of people sometimes refer to the memory response as the secondary response because, of course, it happens the second time. But it'll even happen the third time you see that same pathogen, or the fourth, or the fifth, or the sixth. So it's better to say memory response because, as that name implies, what's going to get activated here is your memory of that first exposure you had, that primary exposure. So the problem with the primary exposure is you've never seen it before, right? You've never seen it before. So it's the first time your body comes into contact with it. So they're scrambling to find naive B cells and naive T cells that recognize this specific antigen. And hopefully several of them do, right, on different epitopes on this antigen. When these cells become activated, they're going to proliferate. So once a B cell recognizes and gets the proper signaling that we'll talk about, it's going to multiply. That same B cell is going to make copies of itself. So you have a whole bunch of B cells that recognize now that specific epitope on that antigen. And then they're going to differentiate even further into plasma cells and actually secrete those antibodies, which are proteins which can travel very easily through our lymphatic and circulatory system, right? Because they're much smaller. They're proteins. They're not a cell. Some of them, though, when we activate, and every time, even if it's a memory response that started this whole thing, some of those B cells don't become plasma cells. They stick around in the lymph nodes and travel to other lymph nodes in other parts of the body, and they're what we call memory B cells. Same is true whenever a T cell is activated. Some of them are going to become what we call effector cells. They're going to go out and do stuff, right? Plasma cell is an effector cell. It does something, right? T cells, some of them are going to become memory T cells, and they're going to hang around, right, and distribute throughout the body. So that the next time you're exposed, you already have cells all throughout the body that recognize that particular pathogen. So what's the benefit to that? Is when you, as the very first time you've seen it, it can take 7 to 14 days before you have substantial response to that pathogen. Because again, we've got to find those naive B and T cells that actually recognize that brand new antigen. That takes time. So that factors into the flu shot, right? Because when they give us the shot, we may, hopefully the whole point of why we're doing it is because we want to know what it looks like in this weakened form, right? We want it to know what it looks like in this non-actively dividing form. So when you get vaccinated, hopefully this is your primary response. So you're going to find what? You're going to find those B cells and naive B cells and T cells that recognize that specific virus that they gave you. So, but, right, the minute you walk out of Walgreens or the clinic, right, are you protected? No. You've got to wait the time for those cells to be found and activated and mount the response, right, and distribute throughout the body. What's the good news? 7 to 14 days later, hopefully you've got B cells and T cells and antibodies that recognize this. So when you come across the real thing, the live actively dividing, right, somebody coughed in your face, you inhaled it, your body already's got this guy's number, right? And the great thing about that is look at how much faster the response is. So primary response, we could wait up to 14 days before we have a significant response. The second time or any other time, we already have memory cells. Those guys are going to be the ones that initiate the response. And because it's memory cells and not naive cells, right, they already know about this guy. And so their response is aggressive and faster. So notice, right, as early as 5 days, I mean, we're off the chart in an immunological response. Does that make sense to you guys? So, my little PA announcement for the day. So I mentioned earlier, and actually, this would be a great one for a post. Post the link to the CDC's website on flu vaccine, right? That could be somebody's post. I just gave you one that you could share with the class, right? But what do you actually have to do for that discussion board? Let's actually look at those instructions. You do have to respond to 2 other people, but you're not just posting a link, right? Oops, did I post the wrong thing? Oh, I moved the date on the wrong thing earlier. I'm all right. That's okay. I haven't graded those yet either, so if you haven't done that yet, do it. But it would have been good for the web pages that you had to read. I got confused. That's why I was like, why is that deadline? Okay. Yeah, this is the one. Okay, this is the discussion board, right? So immune system web link. So I want you to share a web link with the class. It can be anything, a website. It can be a video, an animation, a game, quiz, flat, I don't care. It just somehow has to be related to immunology. Write the title as the first line of your post, right? Then include the web link, right? Highlight the link and click the chain icon and paste the link into the pop-up box to make it a hyperlink on the web page. So I'm going to show you how to do that. Give a description, and so of course 1 point for doing that, of the web link you have provided and explain why you chose to share this site. And then remember you have to look at least 2 other people's. And you're going to write something so I know you actually read what they or watched whatever it is they posted. So remember we click right here to reply. So Adrian, thank you, already posted something. Look, so see? So immunology for kids, right, is her title. She found these web pages interesting, right? This is probably why she chose it. And then she also linked it so we should be able to click on it. And sure enough, look at that. Ooh, a dendritic cell. That's cool. Okay, so for instance, if I wanted to do, but I'll let somebody else actually do it if you guys want. This is what I did this morning. Vaccine. And And the CDC is the Centers for Disease Control, so that's a good, reputable site. Yeah, WebMD's okay. All right, so here is their information for this season. I remember reading more than just this one page, though. Oh, recommendations. So what you would do is copy the link, right? So copy. And then after you write your title, right? So I would write my title, my description, why I chose this page, right? I would paste that in, but then you want to highlight this. Click the chain icon, and you see it pops up. Then you paste it in again, right? And that makes it a hyperlink so that when people click on it, see how it has a little arrow out? It will open up. I'm going to delete mine. Someone else can post that if they want to. That would be a good one to post, right? It's definitely related to immunology.